and then this for my pen. So we're synthesizing fatty acids. What's the starting material? Acetyl-CoA. When we're making ketones, what's the starting material? Acetyl-CoA. When we're making cholesterol, what's the starting material? Acetyl-CoA. And I did bring a pen. Thank goodness. All right. And we're also going to talk about lipoproteins finally too. Just keep mentioning those, but we haven't seen them yet. So the structure of cholesterol, you guys should remember the general structural differences of all of these lipids. Cholesterol is very identifiable because it has 4 rings. So cholesterol is a 4-ring structure. And it does have, cholesterol itself has this kind of hydrocarbon tail out there. But it has so many carbons, it's just basically a bunch of carbons and hydrogens. And whenever you have a bunch of carbons and hydrogens, you're hydrophobic. You don't like water. You don't interact with water. There's this little area over here that would be hydrophilic, but it's just a small portion relative to the rest of it. So because this is such a large structure, most of it's just a bunch of carbons, it is going to be insoluble in water, which makes this guy a lipid. It's hydrophobic. And I want you to focus on something else on this picture, is this fatty acid down here. So this fatty acid, this guy, is when you get a cholesterol ester. So a cholesterol or a cholesterol ester, this is what has to be cleaved when it's digested. So when you consume cholesterol in the diet, this is what that cholesterol esterase is going to take off. And so you end up with when you do this, you end up with a free cholesterol. When you take out that fatty acid, you end up with a free cholesterol and a free fatty acid. And that's what you do when you digest it. It's also what you do when your body uses cholesterol. So whenever you want to store cholesterol, you want to put a fatty acid on it. Oh, I want to be erased. So whenever you want to store cholesterol, you have to put a fatty acid on it. But whenever you want to use it, you have to take this fatty acid off. And so you're just going to use an esterase. And an esterase will help remove the fatty acid.
So you said like the cholesterol ester, that's the part that...
Cholesterol ester, ester is meaning there's an ester bond here, and that ester bond is to put a fatty acid chain in. And so that's why it's called a cholesterol ester, is because you actually make an ester bond when you put that fatty acid on it.
Okay.
But if you don't have the fatty acid on it, if it's just this, then you just have a cholesterol. It's going to be this. Without the fatty acid, it's just cholesterol. With the fatty acid, it's a cholesterol ester.
Okay.
And an esterase is needed to remove that fatty acid. And what is needed to put on the fatty acid, whenever you want to put a fatty acid on something, you're going to use an acyl transferase. So an acyl transferase will put the fatty acid on. An esterase will take that fatty acid off the cholesterol. So an esterase takes it off, an acyl transferase puts it on. And there's going to be a couple acyl transferases we're going to talk about. But they both just put a fatty acid on, and the only reason you put a fatty acid on is when you're ready to store it. When you're ready to store it, then you put the fatty acid on it. If you want to use it, you got to take the fatty acid off. And this is in my slides somewhere. I'm not just saying this. It's written somewhere in my slides. So cholesterol synthesis is where we're going to start. So cholesterol is needed by your cells for a variety of reasons. Cholesterol is needed to make bile salts, you need cholesterol to make estrogen, testosterone, progesterone, you need cholesterol to make vitamin D. And so cholesterol is really important. We do get it in our diet, and we do get it, and we can synthesize it as well. When we get it in our diet, it will go into chylomicrons, but remember the function of chylomicrons isn't to deliver cholesterol. Basically, chylomicrons are going to deliver triglycerides. When the triglycerides have been delivered, then that chylomicron is going to have some leftover cholesterol that might have come from your diet. If you don't eat animal products, then cholesterol is not in your diet. But if you do eat animal products, you will get cholesterol in your diet. And that cholesterol is just going to go straight to the liver from the chylomicron. So the chylomicron is not delivering cholesterol. What's going to be delivering cholesterol is the LDL. So LDL we're going to talk about later in this lecture. But LDL is a lipoprotein, and its job is to deliver cholesterol. And so you're going to take that dietary cholesterol, you're going to load it up and ship it back out again. And technically it's a VLDL, which becomes an LDL. I'll talk about that later. But LDL's function is to deliver cholesterol. And when you do eat a lot of cholesterol in your diet, then you will not synthesize that much cholesterol. If your human machine is working effectively, then when you consume cholesterol in your diet, then you don't have to make it. Your body shouldn't have to make it if it's in your diet. The problem comes in when your body doesn't care how much you're eating and it's over-synthesizing cholesterol. And so we can become, your body can become overly congested with cholesterol. And we'll talk about that in its relation to heart disease later too. But cholesterol in general is not bad. It's good for your body. Your body needs it. You just don't want to make too much. All right, so deregulated enzyme that you need to know for cholesterol synthesis is this guy. You've seen HMG-CoA before, because you actually make HMG-CoA for when you make ketones as well. This is an enzyme. This enzyme, HMG-CoA reductase, is the most important enzyme when it comes to cholesterol synthesis. So I'll show you that when we get to the synthesis slide. And most tissues can make cholesterol because they have all the enzymes to make it, but really it's the liver primarily that's going to be making the cholesterol and shipping it out and delivering it to your tissues. Yeah. Yeah, high levels of LDL are bad, essentially because you have excessive levels of cholesterol that become oxidized and start dropping off inside your blood vessels. And so you can start accumulating these oxidized cholesterols. And LDL becomes a problem too, because LDL will become oxidized itself and start depositing even more cholesterol to tissues. So it's essentially the shipment method. So it's excess cholesterol, and then that cholesterol becomes oxidized, which makes it act kind of like a free radical in the body. So there's a few issues related to the cholesterol, but excess cholesterol will start to harden your blood vessel walls and start creating plaques in your blood vessel walls. And that's why LDL is the bad cholesterol, because its job is to ship cholesterol around the body. And so if you have too much cholesterol, then that's why LDL has a bad reputation. But technically LDL is not bad, it's doing its job, it's just your body's making too much cholesterol for some reason. And that's why we take pharmaceuticals to try to alleviate that. But those pharmaceuticals have side effects. So there's no really good solution for excess cholesterol yet. But we'll talk about that, in a little bit. So sources of cholesterol. So remember, sources can come from the diet. If you eat animal products, you will have dietary cholesterol. The chylomicron will carry that cholesterol from your diet to the liver. Your tissues, if they have excess cholesterol, HDL will carry them not to tissues, but back to the liver. So that's why HDL is called good cholesterol, or the good cholesterol, because HDL's job is to carry excess cholesterol back to the liver. LDL is called a bad cholesterol because it's taking cholesterol from the liver and dropping it off to tissues. HDL is picking it up and bringing it back to the liver so it can be disposed of properly. So we're going to talk, and then once the liver has that cholesterol, it can make bile with it. It can secrete the excess cholesterol as VLDLs, which will become LDL in the blood. I'll talk about how that happens later. And also the liver will make some of that bile. It'll convert some of that cholesterol to bile, which is also important. So we can synthesize cholesterol though, so we do not need to consume it. It is not essential. That de novo synthesis, that means making from scratch. So we can make cholesterol from scratch using what molecule? What's the precursor for making cholesterol? Acetyl-CoA. So cholesterol synthesis. 26 steps for cholesterol synthesis. And I break it down into 3. I want you to know 3 of those 26 steps. And I'm surprised at how many students don't memorize just these 3. Maybe I should give you guys more steps, and then you'll at least learn 3 of them. I will not do that. But there's essentially 3 stages that you do need to recall. The first step is where we make 3-hydroxy-3-methylglutaril-CoA. In other words, this is HMG-CoA. And in fact, I always kind of forget the name. I say HMG-CoA so much, but it's hydroxymethylglutaril-CoA is the full name. How do we make it? We make it using 3 molecules of acetyl-CoA. So we need 3 molecules of acetyl-CoA to begin the process. When we have those 3 molecules of acetyl-CoA, I'll talk about this first. I'll show you the picture in a second. I'll walk through this and then we'll go through the picture. So the 3 molecules of acetyl-CoA come together to make this HMG-CoA. Once we've made this HMG-CoA, we have to reduce it to mevalonate. And I don't want to get you guys get this confused. A lot of students did on the exam. This is not the same thing as malonyl-CoA. Where do we see malonyl-CoA? I'm hearing a few things. It's technically, so it's not carnitine shuttle, it's not from breakdown. This is in fatty acid synthesis. So when we are making new fatty acids, the very first step is we take an acetyl-CoA. I'm just going to go on and review a little bit here. Acetyl-CoA, we add a carbon to it, usually it's in the form of bicarbonate, and we make malonyl-CoA. Malonyl-CoA is that 3-carbon molecule that we end up making. This is the very first step of fatty acid synthesis, and it's catalyzed by acetyl-CoA carboxylase. I won't write that down. But what I'm trying to say is don't confuse malonyl-CoA with mevalonate. Mevalonate is a product made in cholesterol synthesis. Malonyl-CoA is a product made in fatty acid synthesis. So don't confuse those 2 M words. Mevalonate is the reduced product. When you reduce HMG-CoA, you make mevalonate, and this is the enzyme. It's highlighted in yellow here. This is the highly regulated enzyme for cholesterol synthesis. The third step is that mevalonate is converted in a series of steps. So this is where most of those 26 steps come into play. A series of steps, it's going to be turned into squalene, and then squalene is eventually going to be used to make cholesterol. Those are the 3 steps I want you to know. I'm keeping it so simple, focusing you guys on just the very big picture stuff. Here's a textbook picture. Here's 2 acetyl-CoAs. You add essentially another, I'm trying to see how they did this in here. Here we have another acetyl-CoA coming in here, And then you have, so you use 3 acetyl-CoAs. Here's 2 acetyl-CoAs, here's another acetyl-CoA, and so this HMG-CoA, it's written down here, HMG-CoA is made from 3 molecules of acetyl-CoA. And I don't care if you memorize these enzymes. Thiolase, I don't care if you remember that. HMG-CoA synthase, I don't care if you remember that. But I do want you to remember that we start with 3 molecules of acetyl-CoA. We have 2 molecules, 1 molecule, and with those 3 molecules of acetyl-CoA, what do we make? We make HMG-CoA. So we have made HMG-CoA. So here is just very simple picture that will show you the steps of cholesterol that I need you to know. And by the way, this takes place in the cytosol. So here is acetyl-CoA plus acetoacetyl-CoA. So these are the 3 molecules of acetyl-CoA. Acetoacetyl-CoA is 2 molecules already come together, and here's the 3rd molecule of acetyl-CoA. And we use those 3 molecules of acetyl-CoA, they come together to make HMG-CoA. That's the first step to remember. Don't care if you memorize the enzymes. The second step, you do need to know the enzymes. So step 2 here is where we take HMG-CoA and we reduce it. Look what we're reducing it with. We're using NADPH, which we also needed when we did fatty acid synthesis. The reducing power, so basically we're moving electrons from NADPH to this cholesterol molecule, HMG-CoA. We're reducing HMG-CoA to mevalonate. And this is the rate-limiting step. So this HMG-CoA reductase is that rate-limiting regulated enzyme. So that's why this step is important. HMG-CoA is reduced using NADPH to mevalonate. Then you have all these steps in between, and these are about 20-something steps, 20 more steps in here, to convert this mevalonate to squalene. So that is the 3rd step. You don't have to memorize all these other terms in the middle here, but eventually we take this mevalonate using some ATP and a lot of other stuff in order to convert that mevalonate to squalene. And then squalene can be converted to this steroid, which I don't care if you remember, but eventually that's going to be the last step before it turns into cholesterol. And this does require a variety of molecules, glycoproteins, keratines, coenzyme Q. You actually make a lot of intermediates in cholesterol synthesis that you can use in other pathways. But you don't need to know all those details. You had enough details to memorize. Yep.
So you take 3 acetyl-CoA, HMG-CoA, and then NADPH, so that HMG-CoA, and
None. You don't need to know any enzymes between mevalonate and squalene or any of these intermediate products. And there's a lot of them. There's like 20 steps that we're skipping here. Essentially, mevalonate is eventually converted into squalene. The only enzyme I'm requiring you guys to memorize for cholesterol synthesis is HMG-CoA reductase. You should know that when you synthesize anything, there is going to be some energy needed to be put into it. And they do show some ATP up here. There's definitely going to be some ATP required for this, for sure. But where you have the steps for ATP going in, you don't have to remember. 3 acetyl-CoAs coming together to make HMG-CoA. HMG-CoA is reduced using NADPH to mevalonate. Mevalonate in a series of steps is converted to squalene. Squalene in a couple more steps is converted to cholesterol.
what is the NADPH dropping off? Is it the H or both?
It's the H, so there's electrons in that hydrogen. So when when I say HMG-CoA is being reduced to mevalonate, what's happening there are electrons in the hydrogen are moving to HMG-CoA to make mevalonate. And so when NADPH gives away its electrons, it becomes NADP+.
So why does this process not just use
Why doesn't it use NADH is the question, and my answer to that is I don't know. It evolved to use NADPH. I'm sure that there's some molecular, like why we sometimes use NADH to reduce things and why we sometimes use NADPH to reduce things. I don't have a good answer. Molecularly, there must be something about NADPH that enhances that reactivity that NADH does not. But what that exact molecular complement is, I'm not entirely sure. But we could go back to chemistry and probably figure it out. If anybody wants to come to my office to do that, we will. But yeah, I don't have a good answer. But I will say NADPH is often used in the synthesis of a lot of things. So we need NADPH to synthesize fatty acids as well. Other lipids we use NADPH that we don't talk about the synthesis of. Cholesterol we need NADPH. So for some reason when we synthesize the lipids, we usually use NADPH, not NADH. All right, so here's the first step again. HMG-CoA is reduced. So essentially NADH here gives away its electrons. It drops off its electrons. Those electrons are in hydrogens. When you're looking at reduction oxidation reactions, follow the hydrogens. The hydrogens are what's carrying the electrons. And so basically you move those electrons to here. Here's the H's. So you actually move the hydrogens to mevalonate, and now mevalonate is more reduced. It has electrons, and that's what HMG-CoA does. It reduces HMG-CoA to mevalonate. HMG-CoA reductase is the enzyme that catalyzes this reaction. And it is going to be inhibited by cholesterol. So HMG-CoA reductase is going to be regulated. Do I have that next step? Here we go. HMG-CoA reductase. This is how it's regulated. Primary regulation of cholesterol via rate-limited enzyme, HMG-CoA reductase. It is actually inhibited by cholesterol itself. So cholesterol is going to be a feedback mechanism. It's a negative feedback. Essentially, when you already have high levels of cholesterol, you don't need more. And so that's what cholesterol is going to do. It's going to feed back and stop the production of its own synthesis. How it does that is a little bit different than some of the others. So high cholesterol is actually going to turn on a pathway that is going to turn off the synthesis of this enzyme. So it turns off the synthesis. So if we're not phosphorylating, dephosphorylating, anything like that, cholesterol, free cholesterol, is actually going to turn on transcription factors that's going to go into the DNA and prevent the synthesis of HMG-CoA reductase. I have that in the next slide somewhere. But it also has, HMG-CoA also has a short half-life. It's rapidly degraded. So it doesn't stick around very long when it is synthesized. And when there's lots of cholesterol, you will ubiquinate HMG-CoA reductase, or it'll trigger a pathway that causes its ubiquination. Remember from last term, when you ubiquinate something, actually, did we talk about last term? Okay. When we ubiquinate something, you tag it for destruction, tag it for destroying it. And so when you have high cholesterol, it also makes sure that HMG-CoA is ubiquinated, which means it's going to be degraded twice as fast. So in other words, cholesterol itself has two ways to turn off HMG-CoA reductase. One is by turning off its synthesis from scratch, going into the DNA and making sure that's turned off. And it will also turn on a pathway that will ubiquinate it that H and G-CoA reductase, which means it'll go into a proteasome, and remember proteasomes are going to recycle and break down that, whatever's put in it. So in this case, it's going to break down H and G-CoA reductase. So essentially, if you have here, let's say this is H and G-CoA reductase. This is the enzyme, H and G-CoA reductase. And essentially, when you ubiquitinate something, you tag it with this molecule, ubiquitin molecule, and that ubiquitin is recognized by a proteasome. So a proteasome is just kind of this hollowed out protein that will that will grab onto anything that's ubiquitinated, and it'll actually bring it into this proteasome, and that proteasome has enzymes and other molecules in it that will destroy it. But it's just a tag. Ubiquitin is just a molecule, and that tags things. And if ubiquitin is tagging a protein, that means that protein is tagged for destruction. So that's how cholesterol regulates it, but guess what? We do have insulin and glucagon also regulating this one. So in this case, these guys, just like we talked about before, will turn on phosphatases and kinases. So let's think about this. I don't even have to look at my slides. I'll have to think about this, the fed versus the fasted state. In the fed state, or sorry, when do you think we're going to have cholesterol synthesis? In the fed or the fasted state? The fed state. You only want to make cholesterol when you are in the fed state, when you have extra energy. And so I know that that insulin is going to turn this on, because insulin is the hormone that dominates in the fed state. So let me check, make sure that's correct. Yes, insulin activates H and G-CoA reductase by promoting its dephosphorylation, because remember, it turns on a phosphatase, and that dephorylates things. Whereas glucagon is going to inhibit it, because glucagon turns on a kinase, and it's a fasted hormone, and I just know that it's going to make it inactive. But this is the only enzyme you have to remember for cholesterol synthesis. So you do have to know all of those regulatory pathways, but it's the only enzyme you have to remember for cholesterol synthesis. And this is cholesterol synthesis from your textbook. And even this kind of abbreviates it. But here, you do see mevalonate here. Mevalonate is then going to go through a series of steps. There's lots of ATP that's going to be needed here. We eventually get to squalene. And then squalene has a couple more steps, or just one step actually, before it's synthesis into cholesterol. And we do need NADPH here for the reducing power. All right. High cholesterol. This is a big problem in the United States, probably in other parts of the world as well. We tend to have high cholesterol, but our bodies, if you're healthy, our bodies are able to manage this. Our bodies can manage it. So if your body has high cholesterol, you just ate a hamburger or whatever it might be, it will result in a decreased cholesterol de novo synthesis. Again, that's synthesis from scratch, going into the DNA and creating the H and G-CoA reductase and all the enzymes necessary to make cholesterol. So if you already have a lot of cholesterol, this will be turned off. And so this is the rate limiting step. If you do not have, if H and G-CoA reductase is turned off, you're not going to make cholesterol. If it's turned on, you will make cholesterol. So this is the determining enzyme. It's why it's called the rate limiting step. If it's turned on, you'll make cholesterol. If it's turned off, you won't make cholesterol. So if you do have high cholesterol, though, your body will also store it. It won't necessarily excrete the excess. You will store cholesterol in the liver, and that how you store cholesterol is going to be adding. Remember, I told you if you add a fatty acid to the cholesterol. So I don't draw cholesterol, so I just kind of do 4 rings here. 4 rings. This is my cholesterol. Remember, if you add a fatty acid to it. So if a fatty acid is added to that cholesterol, then you're tagging it for storage. And that's what acyl-CoA cholesterol acyltransferase, ACAT. It's an acyltransferase. Acyltransferases, remember I told you what acyltransferases do? They add fatty acids to things. ACAT is a specific acyltransferase for cholesterol storage. And so that's why it's called ACAT for short, it's an acronym, but it's acyl-CoA cholesterol acyltransferase. And that tags cholesterol, creates a cholesterol ester, and it allows it to be stored. Okay, it should not say lipoproteins here. Promotes storage of cholesterol in cells. Sorry about that. There is another one that will promote storage in lipoproteins, and that one's going to be called LCAT. But I guess since I made this mistake, I won't make you tell the difference. But there's ACAT, LCAT. Both of these will promote storage, and we'll talk about LCAT when we get to lipoproteins. But LCAT, ACAT, these promote storage of cholesterol by putting a fatty acid on it. You put a fatty acid on it, you make a cholesterol ester. So you make a cholesterol ester when you want to store it.
Dr. Carter,
Yes.
just to clarify, cholesterol will only go to the liver when it wants to be stored?
No, the cholesterol, any excess cholesterol will always go back to the liver. Some of that will be stored, and some of it will be made into bile. And that's actually a way that the liver can get rid of excess cholesterol, is it can turn that cholesterol into bile for excretion. So all excess cholesterol eventually makes its way back to the liver. The liver will store some of it, and some of it will excrete by turning it into bile.
Okay.
Yeah, cholesterol is a major regulator of its own synthesis, cholesterol synthesis. Yes.
So I've heard before, like if you get your gallbladder removed, like your cholesterol essentially plummets. That's just because it's being pulled from the diet directly for bile, correct?
So if you have your gallbladder removed, what's going on is that, well, I guess it depends on how they do that, but typically you still have, I don't have a good answer to that, because, I mean, I have heard that when you have your gallbladder removed, you have bile being released all the time from the liver.
Yeah.
And if that's the case, then you're having constant removal of cholesterol.
Mm-hmm.
Instead of when you have your gallbladder, you just kind of store it there and you remove it less quickly or in a slower fashion.
Yeah. So I guess in the inverse, like gallbladder disease or dysfunction could be caused by high cholesterol, like just constant
Yes.
filling.
Yes. In fact, I think there's a picture of that.
Okay.
That I'll show you guys. I'm just grabbing my water real quick. Do I have my water? Oh, thank God. No, I made it 3 times. Alright, so if you also have cholesterol, so one thing, if you have excess cholesterol, you will store it, your liver can store the excess. It can also make it into bile and help excrete it. I'll talk about that one in a second here. Step 2, if you have high cholesterol, your tissues are going to do something. They're going to decrease their LDL receptor, and I'm going to have to talk about this a little bit before I get to the lipoproteins, but we'll talk about this with lipoproteins as well. So let's say we have cholesterol in the blood. I'm going to make the blood red here. So we have cholesterol in LDL. So here's LDL carrying our cholesterol. LDL carrying cholesterol, and I'm going to make cholesterol this green here. So if your cells need cholesterol, all they do, here's your cells, they just put out a receptor for that cholesterol. And so when this LDL is coming by, it can actually, so this LDL is swimming by. This receptor is for LDL. It's actually called an LDL receptor. And it will grab onto this LDL and actually internalize it and bring it into the cell. And that's because the cell needs cholesterol. So, but if that cell has cholesterol, it already has cholesterol, what it's going to do is just get rid of this receptor. And so that's what your cells do. If your cells already have enough cholesterol, they'll decrease the LDL receptor. So the LDL receptor is only there for actually for receiving LDL. So it can actually bind here and bring cholesterol into the cell. But if you don't need cholesterol, just get rid of that receptor, and the LDL will just keep swimming on. It won't have to go into the cell. So if your cell, depending on what it is, because all your cells will need cholesterol. Cholesterol is needed for just for the cell membrane of cells need cholesterol. So all your cells need cholesterol. And if they don't have enough, they don't tend to make their own. They tend to get it from LDL. So they just put out LDL receptors in order to bring in that LDL and all of its cholesterol. So if there's excess, because this slide we're talking about high cholesterol. So if your cells already have high cholesterol, they'll just decrease the expression of the LDL receptors so they don't get more. And we'll revisit this when we talk about lipoproteins. If you have abnormal LDL receptors, it can result in this long name, familial hypercholesterolemia, which essentially means you have high levels of cholesterol, and it's genetic. And part of that is some of us do have high levels of cholesterol. We have to be even more careful with our human machines because you have a tendency or predisposition to accumulate more. So it does run in families because some of us genetically have not necessarily abnormal, but different LDL isoforms so that they don't effectively take in or you essentially just end up with higher levels of cholesterol because your cells aren't properly utilizing it. And that's a genetic, so some of us are just predisposed to having higher levels of cholesterol. Just like some of us are predisposed to diabetes or various other diseases. All right, what is this slide about? Okay, I see. This is a picture from your textbook.
She doesn't know who does.
And essentially, this is showing the regulatory pathways. So again, I've written them out in the other slides, but here you see cholesterol. Cholesterol is going to be, it can have, so if you have high cholesterol, this is a feedback. It can actually turn on, you don't have to memorize these factors, but these are like this SRE binding protein. This is the transcription factor it's going to bind to the transcription factors to turn off synthesis. So this is one of the ways cholesterol will inhibit its own synthesis. It'll actually inhibit the synthesis of H&G-CoA reductase. So it's just kind of showing you what I already talked about. This is how it inhibits. It'll go back and it'll turn off this regulatory transcription factor needed to synthesize it. You don't have to know all that detail, but that is how it turns it off using this steroid regulatory binding protein. So steroid regulatory element is going to be needed to turn on the synthesis of cholesterol. Cholesterol will feed back and turn it off. This other pathway over here, this is where cholesterol will increase the ubiquination. So the ubiquination of cholesterol, essentially tagging cholesterol for degradation. So that's how cholesterol will feed back and actually turn off its own synthesis. It'll help degrade H&G-CoA reductase faster, and it'll turn off the synthesis of H&G-CoA reductase. And this other side here is actually showing the role of glucagon and insulin. Insulin will turn it on, and glucagon will turn on a protein kinase that'll make it inactive. So glucagon makes it inactive, insulin makes it active. So there's a lot of stuff going on in the picture, but I wrote it out in all those slides. So any questions about H&G-CoA reductase regulation? Okay, good. So a little bit about the medications. I don't really go into pharmaceuticals, but H&G-CoA reductase inhibitors, there's a lot of them out and about. They're statins that people use in order to try to lower cholesterol synthesis. These are reductase inhibitors. So statins are actually targeting this regulated enzyme. That's what the statins do, that your uncle or dad or parents or what grandparents are probably taking. I know so many people on statins. Essentially, statins work by competing with the substrate HMG-CoA for HMG-CoA reductase. So essentially, people who are on statins will make less mevalonate, because remember that's what HMG-CoA reductase does. It essentially is taking HMG-CoA. Remember what it is actually doing. It is taking HMG-CoA and it is converting it to mevalonate. I'm going to write on this. Mevalonate. That's what this enzyme does. This enzyme converts HMG or reduces HMG-CoA to mevalonate. And what statins do is that they compete for with HMG-CoA, and they will bind to this enzyme instead. So it competes with the substrate HMG-CoA for HMG-CoA reductase. And essentially, if there's competition, you make less mevalonate. So if you don't make mevalonate, you don't make cholesterol. However, the liver, so when the liver senses, so it only reduces cholesterol about 20 to 40%, so it doesn't totally shut down the pathway, but it will decrease the amount of mevalonate. The liver will also do a few things in order to try to reduce. So if the liver senses low cholesterol, trying to figure out how to teach that, sorry. So the liver, if your cells if there's low cholesterol in the liver, because remember the liver is the one primarily making cholesterol and it's the one that ships it out to the other tissues. But if these statins, if someone's taking statins, they have really low cholesterol in the liver, because that's where the statins are working. The statins are going to the liver and that's where they're doing their job. And so the liver has less cholesterol. And essentially when that happens, it will turn on transcription and expression of LDL receptors. Remember I told you what the function of LDL receptors are. Essentially when you have LDL in the blood floating around, you have cells out here that have an LDL receptor and they will take in LDL. And so the liver is going to take in LDL, and that way you can actually reduce cholesterol even more. So I'm saying that statins work in two ways. One, they compete with HMG-CoA for the active site of HMG-CoA reductase, and in that way they prevent the production of mevalonate. And the second way, honestly, this isn't a question I would ask on the exam, but it's just a secondary, it's an indirect way that statins work, because they essentially make cholesterol levels in the liver low, which makes the liver put out more LDL receptors, which means they bring in more LDL, which means you actually clear more LDL, more cholesterol from the blood. So it's a win-win. Okay, I should never say that about a pharmaceutical. But generally speaking, statins kind of work directly by competing with HMG-CoA, and they actually work indirectly by making the liver make more LDL receptors in order to pull more LDL from the blood. But again, doesn't totally shut down the pathway. You will still make cholesterol, and that's why people can still often have high cholesterol even if they're on statins. So it doesn't always work for people. And I'm not sure what the side effects are. I never looked up side effects for statins. I should. But I would imagine there are. Muscle pain, muscle weakness. Let's get some of those from a nurse up here. All right. So, this is just showing the statin. So it's just one type of statin, pravastatin. And you can see it looks very similar. So portions of statins, they look similar to HMG. And so it's going to be able to bind to the active site of HMG-CoA reductase. It actually competes for HMG-CoA to bind to the enzyme. All right, what do we need cholesterol for? So one way we get rid of need cholesterol. So again, you see this 4-ring structure, you should know that we're working with cholesterol here. So this is, when I see this, I know it's a cholesterol. I see these rings, I know I'm working with a cholesterol. And essentially you need this cholesterol in order to synthesize this guy, which is bile. So bile, you have, bile always has a hydrophilic portion and a hydrophobic portion. So here's a very common bile molecule, folic acid is a bile acid. Here's another one down here. Do I want you to remember this? All I want you to remember is that cholesterol is the primary component of synthesizing your bile acids, your bile salts. Cholesterol is the primary component of that. And you do have a hydrophilic and a hydrophobic portion. Bile salts work because remember what micelles are. So when you have a micelle, it's got these hydrophilic portions, the portions that like water, the aqueous, and then you have these hydrophobic parts that don't like water. And that way they can hide all of these lipids in the middle here. And bile salts, so bile salts will need this hydrophilic portion as well as the inner portion here, which is the hydrophobic portions. And so that's why when you make a bile salt, you need both a hydrophilic portion and a hydrophobic portion to that. And that way they can properly transport fat or lipids from your diet, get them to the enterocyte for absorption. Yeah.
So, is this two separate systems where the cholesterol is secreted in bile salts, one and cholesterol is used to make bile salts? That was previously in our notes.
So cholesterol is needed to synthesize or produce them. So I missed kind of that last part. Like,
So it says that free cholesterol is in the bile.
yeah. So you can also secrete free cholesterol in the bile. So not only do we need cholesterol to make a lot of these bile salts, but we can also excrete free cholesterol in the bile as well.
So bile will carry the cholesterol.
Yeah, so you can actually excrete it, and hopefully, especially if you have high cholesterol, that's hopefully one way to get rid of the excess cholesterol. However, we also have receptors that will reabsorb that cholesterol and the bile salts. So we have a recycling of them. So even trying to excrete free cholesterol in the bile isn't always 100% effective. But yes, we can put free cholesterol in the bile, and we can also get rid of cholesterol by turning it into bile salts. Both of them. And so bile salts are always conjugated. I shouldn't say always, but so here again, bile salts, and these bile salts are made from cholesterol, and they usually are conjugated with something. But you don't have to remember that. It's just keep in mind that bile salts there's not one type. There's lots of types of bile salts, but they're all made from cholesterol. And they're used to make that micelle. So this micelle that is here, the bile salts are associated with this. So you have bile salts associated with it. It's also got some phospholipids in it as well, which is why it kind of looks like a phospholipid membrane. But bile salts are incorporated in this as well. So without bile salts, you cannot make this micelle, which means you cannot properly absorb or break down fatty acids or lipids in general without a micelle. Which means you need cholesterol for lipid digestion and absorption. So this slide is showing the enterohepatic circulation of bile salts. What does entero mean? Intestine. So entero means intestine, hepatic means liver. Hopefully you knew that one already. Entero means intestine, hepatic means liver. So this is intestinal liver circulation of bile salts. So essentially the bile duct is going to be secreting that bile into the intestine. So here's the duodenum of the intestine. and these are gonna help with fat or lipid digestion and take-up. And then eventually though, further down, these bile salts are gonna be reabsorbed.
Dang, man.
Oh, it's time for a break. It's telling us whenever there's a break.
Oh.
Yeah, and we do, it's crazy. It's like you missed it yesterday. So.
Hey, how did you know I was doing it? You know what's the scariest part?
You don't even need the email. I would have known.
You would have known. Bro, go open your phone for a second.
Yeah.
Everyone's crashed out. Yes.
Have you ever watched Nolan? Have you ever watched Nolan? Adam Sandler. Can we please watch that movie? Can we please have fun this week?
I am listening.
I've been waiting for so long to have fun.
Oh no, you didn't. Yeah, now we're good. Yes, so when we bring it into the world, now we have she was about to say, yeah, so we break it down, and we got really like that. And we also pull it back up. Exactly. Yeah. Yeah. 2017. Yeah.
Yeah, the second one.
I have a few like sheets. I have like 4 like sweet, but they're also they have like so once they're but sometimes they're small.
I might just label what I
I used to have 20 problems. I used to brag about it. It's my whole
Yeah, I know. I have one now.
Yeah, I don't know.
Oh, you did?
double check.
Like distractions like that. I was just like,
Literally, if I put a picture of Fat Monica up, I had look at Fat Monica. I had this is me and my sister got it at the same time. Take my one shield.
Free cholesterol should only happen.
I want to go to the hospital. I don't got that type of. I'm bringing you.
I don't like lecture.
Most people are thinking it's a fat issue, right? Yeah, I know. But maybe it's because they're like, I understand the science, you know, the heat, like salad, for example. We're in the South, so and then I was also yeah,
I don't know what they I think it might sense.
myself, I'm sorry. Well, they help transport to the experience. Okay, and then
All you can
yeah, the bio I love what you're thinking. I'll be like a latte. That's how I find it. Unfortunately, I don't have yeah, because of my but yeah,
Oh, you like? I've done that with. Actually, I'm asleep.
I mean, it would be great.
I hit them. I hit them.
Yeah, nothing. But yeah, you're thinking the right way, kind of getting more of this background, it might be tease out. Like, I would say it's probably might be to find something that works for me. But I get, I don't have any like
Oh yeah, and then kind of like radio's
you know, I have people strong style. But I mean, like sometimes those green like things like that. I have a friend who doesn't not consume They're more fibrous. The ice lettuce, like they're.
Somebody gets to that level.
I can see
I didn't get to the level of it. Just
You know what I'm saying? Yeah, the one is the cholesterol regulation and the other one is the hormone itself. This is the enzyme is regulated by cholesterol itself in two different ways, and it's regulated by
So like I drew him out like so like daddy I was trying to do this.
it's like I have to pick which one. It's like study go and do like I have to pick two people. The real big ones. It makes sense. What do you really exactly? Some people break off. If you want to breathe, take it out. It's as bad as you want to succeed.
so with this
You want to succeed. As bad as you only ate once a day, bro. Then you skip out. You want to succeed.
reduction scrap.
Listen, this nigga go school, work, home, sleep. Oh, he go work. Yeah, he's transitioning right now. So he go school.
I don't think I can do that.
Happen.
And then the other one
I'm trying to.
is just
I have to go for real now. That's because everybody's schedule is trash.
synthesized, and so cholesterol will trigger a pathway. It'll trigger a pathway that will. It just triggers a pathway that's not pathway. But it triggers a pathway.
Okay, so that's that. So that picture is
I know, there's so much going on.
so but this is the
Ubiquitous pathway. Another pathway.
and then
Which
okay, that's what I
With insulin glucose.
that's what I like
Wait,
everything that we've learned is okay, I learned how to double check.
yeah, I should have put that on there because that's where I'm like, I'm trying to.
It's a lot.
Yeah, and then sometimes she keeps it consistent.
Yeah.
Because yes, I shouldn't put it on there, but yes. Okay.
Okay, so technically the only thing that's gonna like actually
I still like ours. You got an RSUB.
Yeah.
Only in attendance. Then you have to submit it. Like, yes, you like it.
I don't have any.
By the end of the
like
quarter,
week 7 or something.
basically. But he's not gonna grade it, so it's like week 7, week 8. I do know next week.
It might be, bro.
Because I remember somebody asked me in class, well, before we
Hey
started,
guys.
like, Joe went, and he was like, I don't know.
Yeah. I already did quickly. So if you miss quickly, you miss quickly. If you're in here right now, because someone just asked me to extend it to you, just I can't extend it, but, yeah, just send me a really quick email and I'll try to remember to do it when I get home if you just missed it, because there's a couple of students that missed it too. Just for today only. Because I had trouble getting it started there. So if you did miss quickly, just shoot me an email and I'll fix it when I go home tonight or tomorrow. What are we doing? Why is the class like, oh, they're still outside. So good for you. Alright, so what do we need? So again, we need cholesterol for a few things. It has a few functions. One, it makes bile salts needed for fat digestion. You only need bile for fat digestion. Remember, that's to, bile is necessary to help emulsify the large fat droplets and break them into smaller fat droplets. There's no enzymes in bile, but what bile does is makes it more accessible for the enzymes to get to it. And so bile salts need cholesterol for their synthesis. Another function of cholesterol is synthesis of steroid hormones. So here's a few of them. Cholesterol, we start out with progesterone. It's actually the first synthesis. You don't have to remember which comes first. But progesterone is actually used to make the other two. You need progesterone to make testosterone and estrogen. You also need it to make cortisol and aldosterone. So there's a lot of steroid hormones. You cannot make them if you don't have cholesterol. So it is a really important molecule in the body for making your bile salts, for making sex hormones, as well as some other ones. It's something I don't have on here, but you'll see, you saw it somewhere. It's also really important in membrane integrity. So it sits in your membranes and helps make them a little more rigid, because your membranes are kind of this very fluid. Cholesterol will sit in the membrane and help maintain the structure and integrity of cell membranes. So you need cholesterol in cell membranes as well. So hormones, steroid hormones, cell membranes, and bile salts and vitamin D. That's the other one that's really important function of cholesterol. No, vitamin D is made from cholesterol, not progesterone. You don't, yeah, the starting substance. We'll talk about vitamin D synthesis when we get to vitamins. So here you go, gallstones.
Yay.
So this is a gallbladder full of gallstones, which are primarily about 80% cholesterol.
Forbidden nerds.
Oh, no, that's disgusting.
There you go. Came in too soon. You guys should have stayed out there. Okay. Now we're going to focus on lipoproteins. I've talked about them, it feels like, all through this term already, but we haven't talked about them too much. We talked about one lipoprotein, and that is chylomicrons. So just to remind ourselves, chylomicrons are made where in the body? In the intestine, in the enterocytes, specifically the enterocytes make them. And what is their function? Deliver triglycerides or cholesterol? Triglycerides to tissues. To tissues. And I should say deliver dietary triglycerides to tissues. Because we have something else that's going to also deliver triglycerides. Chylomicrons deliver dietary triglycerides to tissues. There's another, it's called a VLDL, it's going to deliver endogenous or body-made triglycerides. But we'll see all this in the lecture. Lipoproteins. Lipoproteins are needed for lipid transport. That's their very broad general function, lipid transport. This is what it looks like. The typical structure. So you have this phospholipid membrane. So all your membranes, even in lipoproteins, will have some non-esterified cholesterol. So again, if it's esterified, then it's going to have a fatty acid on it. And it's going to be stored. So non-esterified fatty acids, the ones that are being used, so this is in the membrane. It's helping with the integrity of this membrane. It's helping maintain the rigidity, the structure of the membrane. However, in the middle here, this inside of here, I put it in red, this is where we're going to have cholesterol esters. So in other words, these guys are the stored cholesterol. Stored cholesterol, remember when you store it, you put a fatty acid on it, and it makes an ester bond. That's why it's called a cholesterol ester. And so you're going to store the cholesterol in the middle here, although every time you see this orange here, that's going to be a cholesterol that's doing a function. It's not being stored, it's actually hanging out in the membrane, helping maintain the integrity of that membrane. So the other things on this lipoprotein, so again, we have phospholipids and we have something called apoproteins. Lipoproteins or apoproteins, and these are also going to be really important components of lipoproteins. There's a few apolipoproteins. Actually, she said, so the student that just took the boards, she said there was a lot of questions on vitamins, the regulated enzymes, and she said lipoproteins. She got a lot on the lipoproteins. So I'm trying to make it more important so you guys pay attention for another 45 minutes or whatever it is. So here you go, lipoproteins. These are the ones, and they're named based on their density. Except for chylomicrons, they're named based on their density. And here's the picture showing you their density. The least dense are chylomicrons. The most dense are HDLs. And the ones in the middle here are VLDLs and LDLs. So VLDL represents very low density lipoprotein. That's what VLDL means, very low density lipoprotein.
So, how microns, are they delivering, fats?
Chylomicrons are delivering triglycerides, fats.
So when they deliver the triglycerides, they get smaller, right?
When they deliver stuff, they get smaller. They get less dense, yeah.
So would allow, how would micron become the LDL?
No, I'm smiling because something like that is about to happen. A VLDL is going to become an LDL. But chylomicrons don't become anything except for a chylomicron remnant. So when chylomicrons drop off all their stuff, they become a chylomicron, it's called a remnant. And that's when it's got just mainly leftover whatever triglycerides and cholesterol that it might have. That's when it goes to the liver. So when it becomes a chylomicron remnant, it goes back to the liver, and the liver is going to take those triglycerides, repackage them, and put them into VLDLs. No. But I will talk about how they are made, actually. But because they all have different functions. So with the lipoproteins, what I want you to know are the apoproteins that are on them and their functions of that lipoprotein. So chylomicrons, their function is to drop off triglycerides from the diet to tissues. VLDLs, their function, just a heads up, you're going to see this in the slides, their function is to drop off triglycerides to tissues as well, but they don't drop off dietary triglycerides, they drop off triglycerides that the liver makes. Remember, we have fatty acid synthesis, we can make triglycerides, and so when we do that, we can repackage those triglycerides, and when we do, we package them into VLDLs. So the function of VLDLs is to drop off triglycerides to tissues. They have the same function as chylomicrons, except for the fact chylomicrons are carrying dietary fats. VLDLs are carrying endogenous fat made from your cells. So not from the diet, they're coming from the liver or some other cell. LDLs, their function is to drop off or deliver cholesterol to tissues. HDL, its job is to take cholesterol from tissues and bring it back to the liver. That's a big picture of what they're all doing, and all the details we'll talk about in the slides. But I do want you to know the density. Chylomicrons are the least dense, and you can see in the picture here, they're really big. Like this is just part of it. They're the least dense, essentially they're just full of triglycerides. So the higher number of triglycerides that they have, the less dense. So the more triglycerides they have, then the less dense they are. More likely they're gonna float, I should say. VLDLs are the second least dense because remember they also have a high level of triglycerides. LDL, so VLDLs actually become LDL in the blood, and I'll talk about that in a little bit, but they're low density. These guys have a high amount of cholesterol. So low density lipoproteins have the most cholesterol of all the lipoproteins. LDLs have the most cholesterol. The high density, the reason high density lipoproteins, so they have the highest amount of protein. They have the most protein associated with them. That's why they're the most dense. They also have a lot of cholesterol as well, not as much as LDL, but between cholesterol and all the proteins, the HDL is going to be the highest and the most dense of all of them. So from least to most, it's chylomicrons, VLDLs, LDLs, and then HDLs.
You have a question, Dr. Furman?
Yes.
Can you go over again what all they transport?
Yes, I'm gonna, that was just the big picture. The slides will actually do that again. Because, I'm just giving the big picture, but you'll be bored in a minute here, because you're gonna know this so well. So chylomicrons are carrying triglycerides. Here's showing you the percentages. So the yellow here is triglycerides. It's got the most. VLDLs have the second most. LDLs have a little. HDL has a little. If you look at LDL next, LDL is the one with the most cholesterol. So cholesterol is blue here. So LDLs have the most cholesterol. You don't have to memorize percentages, but you should know that LDL has the most cholesterol. And HDLs have the most protein. And that's why they have different levels of density. So the most dense, the reason why HDLs are the most dense is because their high amount of protein is what makes them dense. But they also have cholesterol and triglycerides and phospholipids and all that. So this also tells you a little bit about their functions. Chylomicrons, their function is to deliver triglycerides to tissues. VLDLs, their function is to deliver triglycerides to tissues. LDLs, their job is to deliver cholesterol to tissues. And HDLs have a few different functions. And so they're kind of more complicated. One of their functions is to pick up cholesterol, take it back to the liver, but they have a function for these proteins I'm going to talk about in a minute here. So chylomicrons, we'll start with them. They're made in the intestine, the enterocyte, and they carry dietary lipids. So triglycerides, phospholipids, cholesterol esters, all of that will be in the chylomicron. And remember, when they get them from the diet, remember everything's broken down by enzymes. You absorb it in the enterocyte, and then you remake them and package them into chylomicrons. So you actually have the long-chain fatty acids are going to be converted back to triglycerides. Here's an acyl transferase. So remember, acyl transferases are what's going to put the fatty acids onto the glycerol backbone. So you remake the triglycerides, you remake the phospholipids, and you remake the cholesterol esters. All of this, you remake them all in order to package all of those great lipids into the chylomicron, so that chylomicron can take them into the lymph, into the blood, and deliver them to tissue. All right, and just an FYI, just a review, short chain and medium chain are not converted back to triglycerides. They're the only ones that will not go into the chylomicrons. So they can go straight into the blood where they can be carried by albumin to the liver. So short and medium chain fatty acids are treated differently. They're not incorporated into chylomicrons. So something I didn't mention yet is this apolipoprotein B48. So keep in mind, chylomicrons have a lot of triglycerides. They do have some cholesterol, phospholipids, and by the way, they also carry the lipid-soluble vitamins. What are the lipid-soluble vitamins?
A, D, E,
A, E, D, K. So it's also carrying your lipid-soluble vitamins. They do have that phospholipid membrane. Cholesterol is there to help strengthen the membrane. And they also have ApoB48, apolipoprotein B48. This guy is specific, unique to chylomicrons. So an exam question, which lipoprotein has ApoB48? Chylomicrons. It's the only one that has ApoB48. And it's put on in the enterocyte, in the intestine. And the reason it's there is for a couple of reasons. One, well, primarily for integrity. The proteins are going to give structure to the lipoprotein. And so chylomicrons have an ApoB48 that's giving structure to that chylomicron, because remember, phospholipids are loosey-goosey. They're really flexible. They need rigidity. Cholesterol helps lend rigidity, and those apoproteins do as well. So they help create a stronger structure for the chylomicron. But they're also going to be useful for recognition by other cells. So again, it's released into the lymph, then it goes into the blood for transport to tissue. So transported by blood to tissues, it is hydrolyzed at certain tissue sites. Hydrolysis occurs via lipoprotein lipase. Remember your hormones. What hormone turns on lipoprotein lipase? Do you make chylomicrons in the fed state or the fasted state? In the fed state. So in the fed state, insulin dominates. Insulin is what turns on lipoprotein lipase. And lipoprotein lipase is needed in order to pull the fatty acids from the chylomicron and get them into the cell. So adipocytes, how do they get those fatty acids from the chylomicron? They use lipoprotein lipase. So you need lipoprotein lipase in order to pull those fatty acids from the triglyceride and bring those fatty acids into the cells. And it's mainly adipose tissue and muscles. Those are the main ones that are grabbing onto these triglycerides. And that's because they need them. Muscles need it because they use fatty acids to make ATP. And adipocytes need it because they're going to store those fatty acids for use later by the body. So those are the primary tissues that are going to be using this lipoprotein lipase to grab those triglycerides and bring them into the cell. So it releases the fatty acids. That's what lipoprotein lipase does. Remember, lipases, what they do is they take off these fatty acids. So the lipase will pull off these fatty acids, and that way the fatty acids can be brought into the cell. And insulin will activate lipoprotein lipase. I have that somewhere. Chylomicrons belong to a family of molecules called lipoproteins. Each lipoprotein is associated with different apoproteins, so we're going to be defining a few different apoproteins. The lipoproteins are named based on density. We already talked about that. Chylomicrons, are not one of those, but we have LDLs, VLDLs, HDLs. They're named based on how dense they are. And this is kind of the rule of thumb. The higher the concentration of lipids, the lower the density. The higher the concentration of proteins, the higher the density. So that's why HDLs are the most dense, because they have the most protein. Which is all written in my slide somewhere. Why do we need them? We need them because lipids don't like water. So the only way we can transport these is by lipoproteins. So they're very hydrophobic in the core because they're carrying lipids. And essentially they're carrying those lipids and depositing them. Deposition just means depositing or removing. And so the tissues are going in, grabbing what they need from the lipoproteins, And so the lipoproteins are gonna get smaller as the tissues pull what they need from those lipoproteins. However, we'll also talk about plaque formation, because plaque formation also occurs because lipoproteins are accidentally dropping stuff off. And plaques tend to be fatty, there's calcium that can stick to it, so a lot of things can stick to these plaques. And when these plaques accumulate in your arteries, then that's what can cause heart disease, risk for stroke. So lipoproteins will accidentally drop some stuff off, and that will start clogging your arteries. Even if you're healthy, you will, like we just naturally, our arteries, just like a sink or your plumbing system at your house, it has to be replaced sometimes, or Drano or something in order to unclog that stuff. The same thing happens to your blood vessels with these lipoproteins. They start clogging your blood vessels. So there's three things. The lipoprotein, that's the main component. It has the shell that's got protein, it's got phospholipids, it's got cholesterol in it. Apoproteins are those proteins. So when they are associated with lipoproteins, proteins are called apoproteins. So apoproteins are just proteins attached to the surface of lipoproteins. And they have a variety of functions, so we're just going to talk about the apoproteins individually, because there's a few different types of apoproteins. We've already identified one, and that was ApoB48. What is this specific to? Chylomicrons. But there's others that we're going to learn about. And then here's this lipoprotein lipase we just learned about. It's the enzyme attached to cells responsible for bringing fatty acids or triglycerides into the cell. So we're going to start with apolipoproteins and what they do. They do these things. So they are associated with lipoproteins. They provide recognition site for cell surface receptors. We'll talk about that. They can also help activate enzymes. And they do help with structural components. For example, ApoB48 is mainly there to maintain the structure of chylomicrons. But here's all the others. ApoB48, first one, we've already talked about it, unique to chylomicrons. It helps maintain the structure of chylomicrons. That's really the main function of ApoB48. And it is unique to chylomicrons. ApoB100. This one is recognized by LDL receptors. So remember when I told you that when you have LDL swimming in the blood, then you have cells that will have this LDL receptor. And it's going to recognize something on VLDL and LDL. So essentially, they're going to recognize this ApoB100. So ApoB100 is actually what they're recognizing. So ApoB100 is what recognizes the LDL receptors. If a lipoprotein does not have an ApoB100, then the LDL receptor would not recognize it. The ApoB100 is specific to these two lipoproteins, VLDL and LDL. They're the only two. Sorry, HDL, by the way, has most all of them. So I shouldn't say that. HDL will have a little bit too. But the ones that are actually using the ApoB100, so let's just say it is unique to them, is VLDL and LDL. Because they're the ones, well, I haven't told you this yet. VLDL, when it drops, so VLDL is carrying triglycerides to tissues. When it drops off its triglycerides, VLDL becomes LDL in the blood. VLDL becomes LDL. And so VLDL and LDL are kind of the same lipoprotein, except one of them is just dropped. VLDL is the one that's first made, is dropping off triglycerides. When triglycerides are mostly gone, all that's left in the VLDL is LDL. At that point, it becomes LDL. And so that's why both VLDL and LDL have an ApoB100. And that is what's recognized by LDL receptors. ApoA1. This one is specific to HDL. It is important for, so HDLs, by the way, are important for, it's called reverse cholesterol transport. Reverse cholesterol transport. So if LDL is carrying cholesterol to tissues, HDL reverses that. It takes cholesterol from tissues and brings it back to the liver. So that's why HDL is good cholesterol. It's doing reverse cholesterol transport. It's taking cholesterol away from the tissues for its disposal. It's taking it to the liver so that the liver can do something with it. It can put it out into the bile, and you can help try to remove some of that cholesterol that way. It does also activate, this is called LCAT. LCAT. LCAT is just like ACAT. It is an acyltransferase. This AT at the end just stands for acyltransferase. And we already know what this guy does. It puts fatty acids on things. And so if you put a fatty acid on the cholesterol, you store it. And that's important because if free cholesterol is not stored, then you might lose it. And so in order to get it in the middle of this chylomicron, so that it's, or the HDL or LDL, to get it into the middle, you have to put a fatty acid on it. So that's why this LCAT, so APO1 will activate LCAT. It'll activate this enzyme that esterifies cholesterol on the surface of HDL particles in order to store it. So when you esterify cholesterol, you're essentially you're putting a fatty acid on it to store it. And you're storing it in the middle of that. And that's to keep it stable so it doesn't drop off accidentally as the lipoprotein is moving around. And this other part, I'm not, I'm trying, okay, we'll just teach it. The primary mechanism for HDL uptake, so HDL has its own receptor, it's SRB1, which also binds to ApoA1. So ApoA1 is very important to HDL. It does three things. It only does two things. So it activates LCAT, which esterifies. LCAT is just a lipoprotein acyltransferase. And it'll esterify cholesterol in order to store it in the middle of that HDL here. And it will activate SRB1 so that the HDL is more easily recognized so it can be taken up by the liver. The liver has other ways to take up HDL, but this SRB1 is the primary way that the HDL is recognized. And without ApoA1, the liver will not be able to recognize the HDL, or at least by the SRB1 receptor. All right, two more to remember. One is ApoE. ApoE is common to all of them. And it is the one where the liver can take them all up. So all of them have an ApoE, and that is how the liver can recognize all of them. No matter what they're doing, where they are, or not where they are, but if it's a chylomicron, whether it's an LDL, a VLDL, the liver will recognize it because it has an ApoE. HDL, and the liver can take them up. This is actually how chylomicron remnants, so chylomicrons, when they've dropped off everything, how does the liver recognize it and bring it in? It's from ApoE. The liver can pick up LDL and VLDLs the same way. It can use the ApoE receptor recognition. It can also use the LDL receptor for recognition. So it can use either or. But ApoE is common to all of them. It's one of the ways that the liver can recognize them and bring them into the liver. E for everyone. There you go. But again, it's for uptake by the liver. So the liver, that's how it recognizes them and brings them in. ApoC2, I'm going to get to the second part of ApoE in a minute. ApoC2 is really important. It's common to all of them, but it's really most important for chylomicrons and VLDLs. So all of them will have it, but it's mostly used for chylomicrons and VLDL, because it activates lipoprotein lipase. And lipoprotein lipase, remember, that's important for triglycerides. So it's the ones that are transporting triglycerides that will need LPL activated. So ApoC2 is a protein on the lipoprotein that will activate LPL. So ApoE and ApoC2, this one actually is on HDLs. The reason why HDLs have so much protein is that they actually are holding their reservoir for protein, these apoproteins for other lipoproteins. So chylomicrons actually get their ApoE and ApoC2 from HDL. VLDL gets its ApoE and ApoC2 from HDL. So HDL has a second function. It doesn't just do reverse cholesterol transport. Another one of its function is just to act as a reservoir. And so if it sees a chylomicron swimming by, it will give it an ApoE and an ApoC2. If it sees a VLDL swimming by, it will give it its ApoC2 as well as an ApoE. So that's why HDLs have all those proteins. They're actually holding them to give to the other lipoproteins as well. And that's specifically the ones that it's carrying and holding for the others, it's ApoE and ApoC2. So HDL provides it for the other lipoproteins, that's what ApoC2, and ApoE as well. HDL provides it for the other lipoproteins. So those were all the lipoproteins. So just know the function of these lipoproteins and know which lipoprotein has them. Whether it's all of them or whether it's unique to just one of them or two of them. Lipoprotein lipase, this is just review. We know that it's just an enzyme. It sits on the walls of your capillaries, especially if your adipocytes and muscles. They're the primary ones that want triglycerides. They hydrolyze the triglycerides to take off the fatty acids. And then that's how the cells bring in those fatty acids. Without lipoprotein lipase, the cells, your adipocytes and your muscle cells wouldn't be able to bring in triglycerides. So if they do drop them, if they don't take them up immediately, then albumin can grab them, because remember, albumin can carry fatty acids. So it can pick up any excess that might have been left behind. Or it'll just sit on the walls of your arteries and start clogging your arteries. Yeah, sorry. That's very optimistic. All right, regulation of LPL. Remember that does lipoprotein lipase, is it more likely active in the fed or the fasted state? Fed state. And so insulin is going to turn it on. So lipoprotein activity increases in the fed state. That's when you tend to have a lot of triglycerides in the fed state, and so lipoprotein lipase is active in the fed state. And so insulin is going to activate it. So there is like a little area here. I wouldn't try to confuse you on the exam, but when you're fasted, it can become active, and that's only because depending on how you're using your body. So there's gray area there. But generally speaking, it's mostly turned on in the fed state, and it's reduced activity in the fasted state, unless you're using your muscles and you need a lot of triglycerides. But generally speaking, it's turned on in the fasted state, which means insulin is going to turn this guy on. Chylomicrons start out as nascent. What does that mean? They're new. They don't have all of the life skills yet to do what their function is. In the blood, HDL has to provide the chylomicron with ApoE. Again, that's for recognition by the liver. And ApoC2, which activates LPL. So that's just a repetition from an earlier slide. What is the function? The function of chylomicrons is to deliver triglycerides to tissues, primarily muscles and adipocytes. Muscles are going to oxidize the fatty acids for energy. Adipocytes are going to store those triglycerides. They're going to store the fatty acids as triglycerides. So that's the function of chylomicrons. Once all of those triglycerides, or most of them, are gone, what's left is a chylomicron remnant, because it's lost a lot of its lipids. It might have still cholesterol. If you've had cholesterol in your diet, if you're a vegetarian, you don't eat any animal products, then you won't have this. But if you do have cholesterol in your diet, then the loss of triglycerides, you'll have low triglycerides, you might still have cholesterol, but the liver is going to remove the chylomicron remnant, and it needs this ApoE in order to recognize it and to remove it from the blood. And it's actually going to be recognized and endocytosed. So endocytosis is how that chylomicron remnant gets into the cell. And this is just the picture showing everything I've talked about. So here you have chylomicrons being made in the small intestine. It goes out into the blood. HDL is going to give it C1 and ApoC2 and ApoE. It's going to go drop off tissues via the lipoprotein lipase. It's going to free those fatty acids from it. And eventually, it's going to become a chylomicron remnant, and it's going to be taken back up by endocytosis into the liver. VLDLs. Very low density lipoproteins. These are made in the liver, not the intestinal cells. And they have their own specific apoprotein, ApoB100. This apoprotein is important for recognition by LDL receptors on cells. So VLDL is made in the liver, and it's going to be secreted directly into the blood. Its function is to carry endogenous triglycerides. Endogenous is not dietary. Endogenous means made within, so it's inside. It's the triglycerides made inside your body, versus the other word would be exogenous. Exogenous means from your diet. You get it from outside in. So exogenous versus endogenous. Endogenous means you make it inside your body. Exogenous would be like dietary. You get it from somewhere outside. So endogenous triglycerides are going to be what the VLDL is carrying, and it's going to carry them from the liver to the tissues. So the function of VLDLs is to carry triglycerides from the liver to the tissues. And then you have that lipoprotein lipase. It's going to pull out those fatty acids. Lipoprotein lipase, just like with chylomicrons, is going to pull off the fatty acids from the triglycerides in order for the cells to get those fatty acids into the body, into the cell. When VLDL drops off its triglycerides, it is eventually converted into LDL in the blood. In the blood, VLDL becomes LDL. There is an intermediate, an IDL. You don't have to really worry about the intermediate, but there is an intermediate that's made in between there. LDLs, once we make an LDL, the function of an LDL is to transport cholesterol to tissues. So VLDL, its function was to transport triglycerides to tissues. LDL, its function is to transport cholesterol to tissues. And this is when the LDL receptor becomes into play. It's going to bind, cells will bind to the LDL receptor, and they're going to recognize the LDL because LDLs have an ApoB100. That ApoB100 was put on it in the liver. So it's got this ApoB100. That's what the cells will recognize, and that's how they're going to bring LDL in and get the cholesterol from LDL. And this is going to be endocytosis. It actually brings the whole LDL into the cell. And once the LDL is taken up, essentially it's going to be, it's going to form endosomes. Lysosomes are going to be, I call them the recycling center of the cell. So the LDL is just going to fuse with the lysosome essentially, and the lysosome will help break down the LDL and pull out the parts that are needed, like the cholesterol. That's essentially what this is saying. So the LDL is just going to go into that lysosome in order to get the free cholesterol out of it. And now that cell can use that cholesterol to make whatever it needs. It might need vitamin D, it might need to make estrogen, whatever the cell needs to make. And there's a lot of molecules that we can make from cholesterol. Or it just wants to put that cholesterol in the cell membranes in order to solidify and help the cell membranes be more rigid. So here's the VLDL. The VLDLs are put out into the blood, and remember they have an ApoB100 on them, but they get the ApoC2 and the E from the HDLs. They're going to drop, VLDL is going to drop off those triglycerides to tissues. Eventually it's going to become an LDL, and that LDL still has the ApoB100 on it. Other tissues are going to be taking up that cholesterol. Anything that's left, if there's any LDL left in the blood, it will eventually make its way back to the liver, and the liver can take it back up. So LDL is made in the blood from VLDLs. As VLDLs drop off their triglycerides, they end up with a higher concentration of cholesterol, they become less dense, eventually becoming LDL. So again, the job of very low density lipoproteins, here you see all the triglycerides. Once the triglycerides are dropped off, then you essentially end up with mostly, this is high density lipoprotein. What is this about? Transfer, they can also give, so high density lipoproteins will actually even give the VLDL cholesterol. So HDL will actually give more cholesterol to the VLDLs, helping change them to LDLs. So HDL will give VLDL cholesterol too, changing the VLDL in a faster way to LDL. And remember, which lipoprotein has the highest amount of cholesterol? LDL. LDL has the highest amount of cholesterol. HDLs have cholesterol, but they don't have as much as LDL. They have more protein, is HDLs. You can't leave yet. Okay, let's see if there's anything else that I really need to show you guys. All right, we're going to pick this up next week. So the videos are posted for this, so it's good to watch the videos. Well, at least in the protein. The last part of next week, there's amino acids. I'm going to be shifting those. So again, I'm not stressed out about the time, so I don't want you guys to be. I'm having to shift this class a little bit from last term. But yeah, let me know if you have questions. There's not much left.